Hello, everyone. Welcome to Pocket Size Bio with Dr. Casco. This is part 3 of the gene expression or protein synthesis module, and we're going to talk about translation and protein synthesis. Okay, so let's get to it. So protein synthesis, basically translation, occurs in the cytoplasm with the ribosomes. Okay, and this is referred to as being RNA-directed synthesis of a polypeptide. And the reason why it's RNA-directed is because you use RNA as the template to create the protein from the amino acids that are starting to chain up depending on the particular sequence of the mRNA, right? That just came from the nucleus. So if we take a look here, right, this is the essential setup of how translation works. Basically, you have your ribosome, right? And this ribosome is reading, okay, the mRNA. And as it's reading the mRNA, it's bringing, okay, tRNAs to come in and transfer an amino acid that each of them carry, right? In a sequence, right? So more tRNAs come, they provide a specific amino acid, and these amino acids will start to chain up to create a growing amino acid chain, basically a polypeptide, right? So we're going to see this in a lot more detail. So first, we need to talk about what are the types of RNAs that are involved in translation and protein synthesis, right? So you have mRNA, which we know already, right? That mRNA transcript that is coming from the DNA template of a particular gene. And there's some other ones that we need to know. tRNA, which is referred to as transfer RNA. These are small little molecules. Here you can see one little example. Here are some other examples of the diagrams representing them, right? They carry specific amino acids depending on their particular anticodon sequence. So every tRNA has its own little individual triplet, right, nucleotides or anticodon that will define what type of an amino acid it's carrying. Okay, so if you take a look here, CCG, this particular tRNA is carrying glycine, right? AAA carries phenylalanine, and so forth and so forth. So there's many, many different types of tRNAs, and we're going to be taking a look at how they come in and provide the amino acid to the chain, growing chain, right? And then you have rRNA. rRNA is referred to as ribosomal RNA, and it's a sequence of DNA that gets transcribed into this particular RNA to make physically part of the ribosome, right? It doesn't produce a protein. It doesn't have information to create a protein. It stays as a RNA, right? And it actually becomes part of the ribosome, okay? Now, in terms of rRNA, that's really what you need to know for the WACE program, okay? It doesn't go beyond that level of detail, okay? So that's the general gist of translation right now, the key components that we need to know, right? In addition, the ribosome actually has little grooves, particularly 3 little grooves in which the tRNAs can come in and kind of snap in, right? There's 3 parts to that, but again, this is beyond the scope of the WACE biology course, right? But I will mention it every now and then. That's why I kind of am depicting it here. All right. So let's take a look at it in more detail. So this whole idea of translation occurs in 3 stages, right? You have initiation, you have elongation, and then you have termination, right? The first one, initiation, basically occurs whenever you have the ribosome coming in and latching onto the mRNA from a 5' to 3' direction, right? So this would be 5' here, 3 prime, and the ribosome will come in and join and kind of ride it and read it like a roller coaster throughout the sequence. Okay. And ribosomes read it in sets of 3, right? Basically the codons that I mentioned before. So these codons basically will not bond, but they'll attach to the corresponding anticodon of the tRNA. Okay. And for the protein to actually start, for the amino acid sequence to actually start being made, there's a very special sequence, or there's a very special codon that needs to be first read, which is AUG. It's referred to as the start codon, and this codon codes for the amino acid methionine, M or MET, depending on the genetic code that you're using, right? And this signals the start of translation. And then the ribosome will continue, and there's actually supposed to be another tRNA that comes and couples together with the first methionine, right? But again, these are subtle little details beyond the WACE program. So you have this start codon, this tRNA comes in with its corresponding anticodon. In this case, it's UAC, right? It'll come in and bind, and it brings along this amino acid, methionine. All right. So this is what I mentioned before. 2 codons enter at the beginning of the ribosome, right? But afterwards, individual single codons enter one by one that we're going to take a look. Okay. So the second step, elongation, once you have these first additional tRNAs, right, you have the ribosome continuously reading along from codon to codon, right, from a 5 to 3 prime direction. And they would bring, depending on the particular sequence, that corresponding amino acid. Okay. And of course, the main function of that ribosome is to cohere everything together, the mRNA with the tRNA, and enable a passing of the growing amino acid sequence, right? So what happens, what I'm referring to that is that as you have this amino acid that is growing, this little strip will detach from this current tRNA and bind to the following one, to glycine in this case, and everything will shift out, right? tRNA, the one that already gave up its amino acid, will leave, and the following 2 will kind of shift in the convoy line, right? This one will come here, this tRNA will come into the middle portion, and then a new one will come in. And think about it as like passing down the flag, for example, as you go down a convoy. One enters, the other leaves. The middle guy is holding the growing strands, right? And they're passing along the growing string of amino acids, right? These little grooves have particular sites. This is called the A site. This is the P site, right? And this is the E site, the exit site, right? But just know that they're constantly in motion. One comes in, one comes out, and they all flow and take each other's position, right? This is referred to as elongation. So this will continue on, okay, in one direction, right? And until, you reach the end of the mRNA sequence and you reach termination. Okay, what happens here is that you reach a very special codon called the stop codon, right? And there's different kinds of codons that would code for a stop, right? UAG, UAA, and UGA, right? They don't code for a particular amino acid nor a tRNA. Instead, they actually code for a particular molecule referred to as a release factor that will come in and bind to this particular sequence, this codon, and it will tell the tRNA, that last tRNA, and that polypeptide, all right, that amino acid chain that is growing, to basically snap off everything has been finished, and now the ribosome, the tRNA, the polypeptide, um, everything can basically disassemble, right? That marks the end of translation. Okay. Generally, it's near the end of the mRNA sequence. And now this free polypeptide could be, it could go on to get modified to fulfill its 3D conformation, right? And it could be used inside the cell as a signaling factor, let's say, or a structural protein, or it could go out, into the somewhere else into the cell, for example, a hormone, right, to do some other signaling in another cell, right? And just a little bit more detail there. Amino acids, once they're done being formed, their chain is being formed, right? They have particular structures and levels of structures, right? They're referred to as primary structure, secondary structure, tertiary structure, and quaternary structure, right? When you refer to as the chain of amino acids, you're basically talking about just the order of amino acids, right, that are in sequence. It's referred to as a primary structure. Okay. Depending on how unique the sequence is and the different types of amino acids being used, they can start interacting with each other, right? Let's say like phenylalanine with proline, and they can start creating bonds, hydrogen bonds, for example, and they can start creating a 3D shape, which could be a little spiral, which is called the alpha helix, or they can make these flat conformational shapes. These are called beta sheets, right? Beta pleated sheets, more specifically, which is basically the molecules, I don't want to say entirely flat because they are kind of bended at an angle, right? Going upwards, downwards, upwards, downwards. But ultimately, throughout this chain, you're going to have these little conformations, right? These little structures there. The tertiary structures, whenever you have the entire sequence, right, having the particular alpha helixes and beta sheets all together and interacting with each other to create a formal 3D structure. At this point, you can refer to this one maybe to a protein as a protein, right? But generally, proteins actually work with other long polypeptides, right, to create a larger complex, right? And this is referred to as a quaternary structure, where you have a basically coupling of several parts of several different proteins, right, working together. An example of that would be hemoglobin, for example, right? So you have primary structure, secondary structure, tertiary structure, and quaternary structure. Okay, these are more examples, diagrams of what I'm mentioning right here. You can see the alpha helix, where you have um, hydrogen bonding, right, occurring between different amino acids, right, from a carboxyl group to an amino group. Or you can have, depending on the different types of amino acids, you can have these beta sheets. Okay, so if you can see it in a 3D conformation, this one's going upwards. This particular amino acid is going upwards, declining downwards, and so forth and so forth. But ultimately, you have this kind of interaction throughout the whole sequence of amino acids, and you can get this particular conformation. This is a very nice representation, or a 3D modeling representation of aquaporins, right? Aquaporins is a channel protein that is on the cell membrane to enable water to move throughout, right? If we were to look at it in a 3D perspective from a side view, you would see all these alpha helixes. From a bird's eye view, from a above view, it would look something like this, right? So this would be an example of the 3D conformation of a protein. Okay. So hope that helps in clarification for translation. This finishes the 3-part explanatory videos of protein synthesis.